Okay, so hello everyone. If you don't know me, my name is Dena. I'm a second-year graduate student in the Legal Lab. So I'm just kind of getting started with my project. So today I'll talk a lot about the background and preliminary data leading up to what I'm planning on doing during my time here. So my project is focused on the role of microglial D-Serine in synaptic damage following traumatic brain injury. So just a little bit of background on traumatic brain injury. Traumatic brain injury occurs when there's damage to the brain through different mechanisms, such as the direct impact to the head, rapid acceleration and deceleration of the head, as well as blast waves, which are most common in military settings. There is an estimated 69 million cases of TBI worldwide, with an annual 1.7 million cases in the US. Traumatic brain injuries most commonly affect young children as well as elderly populations due to falls, but are also, there are high rates in young adults as well due to mostly motor vehicle accidents. So there's obviously a wide range of injury type and severity that can be acquired, resulting in a wide range of symptoms, including cognitive impairments such as learning and memory deficits or problems with attention, motor symptoms, as well as behavioral changes, such as increased aggression or a higher risk for depression and anxiety. So there are many diverse mechanisms of traumatic brain injury, and we can typically separate this into the primary phase, which occurs directly at the time of impact and involves things such as damage to blood vessels, compromise of the blood-brain barrier, direct tissue damage, as well as axonal shearing. And since this occurs acutely at the time of impact, there's not much that can be done therapeutically to address primary injuries. So we're mostly focusing on secondary injury mechanisms when we talk about therapeutically targeting traumatic brain injury. And these mechanisms occur minutes, days, and can last weeks or sometimes even years after the primary insult. And this involves things such as ischemia, edema, glutamate release, which results in excitotoxicity and mitochondrial damage and oxidative stress. And the aspects that I'll be focusing on the most for my project are synaptic damage and gliosis. So before I talk about the importance of glial cells after injury, I just wanted to emphasize their role under physiological conditions. So we typically think of the tripartite synapse, where astrocytes form contacts with the pre- and postsynaptic side of neurons and are very important for buffering glutamate to prevent excitotoxicity, as well as releasing gliotransmitters that contribute to synaptic transmission. Microglia are also highly implicated in physiological conditions, where they are constantly making contact with neurons and receiving signals from neurons to keep them in a homeostatic state under physiological conditions, such as through the CX3-CR1 receptor. Microglia are very important for synaptic pruning, especially during development, and abnormal microglial synaptic pruning has been implicated in many disease states such as Alzheimer's, schizophrenia, and autism spectrum disorders. Microglia have been shown to be able to contact neurons in an activity-dependent manner and are important for regulating neuronal synchronization as well. And many gliotransmitters released by glial cells are implicated in synaptic plasticity, which for my project, I'll be focusing on synaptic plasticity in the hippocampus, which is mediated by NMDA receptors. And NMDA receptors are composed of multiple subunits, including an obligatory GLU-N1 subunit, as well as GLU-N2 subunits, which can be composed of different subtypes, which I'll talk about a bit later in my talk. But in order for NMDA receptors to be activated, they require three things, such as glutamate binding, a modest depolarization, which is typically due to AMPA receptor activation, as well as binding of a co-agonist, which in the hippocampus is the amino acid D-serine, which I'll be focusing on. And if all three conditions are met, this results in calcium influx, leading to long-term potentiation and gene expression changes, as well as under cases of injury, it can lead to excitotoxicity. So I'll be focusing on the co-agonist for NMDA receptors in the hippocampus, which is D-serine. And D-serine is an amino acid that is synthesized from its precursor L-serine. L-serine is produced in astrocytes as a product of glycolysis, where this PHGDH is the first is the enzyme that starts the first committed step of L-serine synthesis. And L-serine can then be exported from astrocytes into neurons, where it is converted into D-serine by the enzyme serine racemase. And then it's released at synaptic sites to contribute to the normal synaptic functioning of NMDA receptors. So since D-serine is so important for synaptic plasticity under normal physiological conditions, our lab is interested in looking at the role of D-serine under pathological conditions. So to study this, we use a controlled cortical impact model, which is shown here, where a highly pressurized piston makes contact over the direct dura to deliver an impact. And as you can see here, compared to sham conditions up top, there is severe cortical loss in the CCI model. However, the hippocampus remains intact. And when looking at the number of new and positive cells in the hippocampus following the CCI, we see no changes in our model. However, there is a significant decrease in the number of synapses in the CA1 area. So this allows us to study synaptic damage in the absence of cell death, which is advantageous to be able to study remodeling after injury, specifically in synapses, without the confounding factor of cell loss. And on a functional level, you can see here, there is abolished LTP following a CCI injury, shown in black, as compared to wild-type mice in white, in sham mice in white. And this also corresponds to behavioral deficits as well on a fear conditioning paradigm. And I'll talk about the details of this a little bit later, but this is a associative learning task where mice are placed in a specific chamber and receive an aversive stimulus, in this case, a foot shock. So increased freezing corresponds to an increased ability to associate the context with the aversive stimulus. So as you can see here in black, the CCI mice have significantly reduced freezing, suggesting an impairment in learning and memory. So since D-serine is implicated in synaptic plasticity under physiological conditions, wanted to look at if the expression of serine racemase, the enzyme that synthesizes D-serine, is altered after injury. So you can see here in a sham condition, serine racemase in purple is localized mainly to the pyramidal layer of the hippocampus, which contains neuronal cell bodies. However, 3 days after injury, there is decreased expression in this layer and more diffuse glial expression, which when looking at colocalization with IBA1, a microglial marker, we can see increased colocalization of serine racemase with microglia after injury. And so we quantified this using flow cytometry. And as you can see, after injury, the percentage of microglial cells that are positive for serine racemase is significantly increased. And in addition, the mean fluorescent intensity per cell is increased as well. So not only do more microglial cells express serine racemase, individual microglial cells express a higher level of the enzyme as well. So this led us to the question if microglial D-serine is implicated in causing synaptic damage after injury. So to look at this, we use two different mouse lines where serine racemase is conditionally knocked out in microglial cells specifically. And mice receive either a sham or CCI injury. And then in the 5 to 7 days after injury, will undergo behavioral testing, which in this case is the fear conditioning test. And then at 7 days after injury, we harvest the brain tissue for Golgi analysis. And Golgi staining is a silver-based stain, which stains the cell bodies, dendrites, and axons of neurons. So it allows us to visualize individual dendritic spines and quantify dendritic spine density in particular cells in the hippocampus. It also allows us to study the morphology of dendritic spines. So what we're interested in our studies are these mushroom-shaped spines, which are hallmarks of excitatory mature synapses. And we're also interested in thin spines, which, as you can see, have a much smaller surface area and are indicative of degenerating spines. So when looking at the spine density in control serine racemase flox mice, we observed a significant decrease in the density of dendritic spines. And this corresponds to a significant decrease in the percentage of mushroom-shaped spines, as well as a significant increase in the percentage of thin degenerating spines. However, in our microglial knockout mice, this effect is abolished, where we don't see this reduction in spine density, and also we don't see any changes to morphology after injury. So in order to correlate this to a functional outcome, we perform this fear conditioning assay, where 5 days after injury, mice are habituated to a chamber, and the following day are once again placed in the chamber and receive a foot shock coupled with a auditory stimulus. And then 24 hours later, the mice are once again placed in the chamber and assessed for freezing behavior, where freezing behavior is considered to be a fear response. So in this case, a higher percentage of freezing corresponds to a greater ability to a greater learning and memory, as mice are able to associate the context of the chamber with the aversive stimulus. So as you can see here in our control mice, 7 days after injury, there is a significant decrease in the percent of freezing. However, in our microglial knockout mice, this effect is abolished, suggesting that microglial serine racemase ablation is sufficient to rescue learning and memory on a functional level as well as a morphological level. So just to kind of summarize what we know so far, in naive or sham conditions, serine racemase is expressed predominantly in neurons, where it can synthesize D-serine from L-serine, and then release this D-serine at synaptic sites to contribute to normal synaptic function and plasticity. However, in a TBI case, neurons downregulate serine racemase expression, whereas microglial cells, as well as astrocytes, which I'm not focusing on for my project, but our lab has previously also implicated in this D-serine mediated damage. And this glial release of D-serine contributes to damage of and elimination of spines after injury. However, this mechanism of how microglial D-serine is contributing to the synaptic damage after a TBI is still unknown. So this is what I'll be focusing on in my project, where I'm interested in elucidating the mechanism of microglial D-serine mediated synaptic damage following a TBI. And my overall hypothesis is that microglial D-serine mediates synaptic damage by hyperactivation of extrasynaptic NMDARS receptors, which are enriched in the gluN2B subunit, and as well as possibly altering microglial responses after an injury. So the first part of my project will be focused on this gluN2B subunit. The second part will be looking at alterations to microglial responses, such as excessive pruning or changes in inflammation after injury. And then finally, I'll be taking a translational approach to see if some of these components that we found in our mouse model are upregulated in human TBI patients as well. So focusing on the first part of my project, which is to look at whether D-serine mediates hippocampal synaptic damage through hyperactivation of extrasynaptic NMDA receptors, where I hypothesized that the prolonged release of D-serine by microglia acts predominantly at extrasynaptic sites to hyperactivate this gluN2B subunit and lead to synaptic damage. So this gluN2 subunit contains two main isoforms in the hippocampus, the gluN2A subunit, which is predominantly expressed at synaptic sites and is involved in pro-survival signaling as well as synaptic plasticity changes. And then the gluN2B subunit is predominantly expressed extrasynaptically, where its hyperactivation in cases of ischemia, as well as epilepsy and other disorders has been implicated in inhibition of pro-survival signaling and activation of cell death pathways, as well as inhibition of synaptic plasticity. So here on the right, this is a quantification from a previous paper in the lab that looked at the expression of this N2B subunit after injury and found that it was significantly enriched after a CCI in wild-type mice. So to investigate whether this is due to the release of microglial D-serine, I'll be examining some of the signaling pathways of this Glu-N2B subunit. Or I'll be evaluating expression levels of the different components of NMDA receptors, as well as the presence and activation of this kinase, death-associated protein kinase 1, which in models of ischemia, Huntington's, is known to phosphorylate the Glu-N2B subunit to increase its conductance and expression at the plasma membrane, contributing to increased calcium influx and excitotoxicity. I'll also be looking at some of the key components involved in synaptic plasticity, such as pCREB and pERK, as well as BDNF, which after Glu-N2B extrasynaptic activation are known to be downregulated. So I'll be looking at whether there is a difference in expression of these subunits after injury in control, as well as serum racemase knockout mice. Where here I expect that the phosphorylation and expression of the Glu-N2B subunit, as well as DAPQ1 expression, will be increased after injury, while pCREB, pERK, and BDNF will be decreased in our control mice. And I expect that this effect will be attenuated in our microglial knockout mice. And another interesting avenue here would be to see if these same signaling pathways are implicated in astrocytic knockout mice, where serum racemase is ablated in astrocytes, since our lab has previously implicated the cell type in synaptic damage as well. So next, I'm interested in taking a pharmacological approach to see if pharmacologically inhibiting the Glu-N2B subunit can block the synaptic damage mediated by D-serine after injury. So as a preliminary study, I used the inhibitor Ro-maliate, which is a Glu-N2B specific inhibitor, and using ASLET osmotic mini pumps, which connect to the brain via a cannula to deliver this, can deliver the drug at a constant rate over the course of 3 days. So I delivered 3 different concentrations of this inhibitor for the course of 3 days after either a sham or a CCI injury. And to start, looked at some Golgi analysis. So what I found was that, as you can see here in black, this is mice that received a vehicle saline infusion. You see a significant decrease in the percentage of mature mushroom-shaped spines. And this effect was abolished in our mice that received the Ro inhibitor at all 3 concentrations. And a similar effect was seen with the thin spines, where there's a significant upregulation of degenerating thin spines in our vehicle condition, which is once again rescued at all 3 concentrations of the inhibitor. And these are just some representative images where you can see that there is a greater presence of these mushroom-shaped spines in our sham conditions as compared to CCI conditions, where you see more prominent thin spines. And this effect is reversed with application of the inhibitor. So to look at this further, I'm now working on looking at this on a functional level as well to see if this Glu-N2B inhibition can rescue learning and memory behavior, as well as synaptic damage at 7 days post-injury, where I'm using either infusion of vehicle, the Glu-N2B specific inhibitor, as well as DAP5, which is a non-selective NMDA receptor inhibitor. And here I expect that the specific gluin2B inhibitor will rescue both fear conditioning behavior and spine density at 7 days post-injury. And I expect less of a therapeutic effect with the non-selective inhibitor, as this will block synaptic activity as well. And then finally, I'm planning to use a genetic model where the gluin2B subunit is knocked out specifically in excitatory neurons in the hippocampus. So first, I'll start out using the fear conditioning paradigm as before, followed by the Morris-Water maze as a secondary measure. And then finally, I will perform electrophysiology to look at if long-term potentiation is preserved with this gluin2B knockout. And here I expect that the knockout mice will have reduced synaptic damage and improved learning and memory following an injury. And since the gluin2B subunit is also implicated in synaptic functions in physiological conditions, an interesting alternative would be to use a gluin2A knock-in model, where gluin2B is replaced with the gluin2A subunit. This way, synaptic levels of NMDA receptors are not affected. It's simply this gluin2B specific signaling that is abolished. And it would also be interesting to look at whether microglial NMDA receptors are also being activated by D-serine and seeing if ablating NMDA receptors specifically in microglia can also rescue synaptic outcomes in mice. So the second part of my project is then focused on altered microglial responses after injury, where I hypothesize that the hyperactivation of NMDA receptors by D-serine will lead to the complement-mediated tagging of dendritic spines for removal. And this complement-mediated mechanism has been implicated to be important for pruning excessive synapses in development, especially in the visual system, as well as in abnormal synaptic pruning in Alzheimer's disease, West Nile's disease, and epilepsy. So C1q is the initiator molecule of the complement cascade. And recently it's been shown that C1q can interact with phosphatidylserine, which is an apoptotic marker that is externalized in dying cells to signal phagocytosis. However, phosphatidylserine externalization has also been implicated in individual synaptic components, where it leads to elimination of spines without cell loss. So in addition to binding phosphatidylserine, C1q can catalyze the cleavage of C3 into C3b, which is unopsonin, and also signals through microglial receptors for the phagocytosis of synapses. So this, the combination of these signaling pathways would lead to microglial engulfment of specific synapses, leading to the elimination of the synapse. So just to kind of summarize our working model so far and how I see these mechanisms coming together in injury. Here on the right, under normal sham or naive conditions, serine racemase is expressed predominantly by neurons and is released via amino acid transporters to act at synaptic NMDA receptors and is important for normal synaptic functioning and synaptic plasticity. However, in a TBI model, microglial cells will upregulate serine racemase, and they release this predominantly on extrasynaptic NMDA receptors, leading to phosphorylation and increased influx of calcium. And this can lead to the externalization of phosphatidylserine, this phagocytic marker, which is then recognized by C1q. And together, these components can tag synapses to be eliminated by microglia. And a receptor that we're specifically interested in is GPR56, which has recently been implicated in microglial pruning via this phosphatidylserine and complement-mediated mechanism. So some preliminary findings show a significant increase in C1q in wild-type mice following a CCI, which is not seen in our microglial knockout mice. And here with IHC, we see a similar effect where there is an increase in C1q in green after injury in control mice, which is once again abolished in our knockout mice. And when looking at GPR56 expression, I didn't see any significant differences at 3 days post-injury. However, there does seem to be a decrease in GPR56 expression in our knockout mice. So I'll be interested in evaluating this at later time points. And to further evaluate this, I'm planning on examining the co-localization of these complement related proteins in synaptic pruning. So seeing if the proteins C1q, C3, as well as the GPR56 receptor co-localize with synaptic spines after injury. And here I'm expecting to see an increased co-localization with dendritic spines in the CA1 region of the hippocampus in our control mice following a CCI, but not in the mice where D-serine is ablated in microglia. And we also can't exclude the role of astrocytes in this complement-mediated synaptic pruning, as astrocytes have also been shown to be able to phagocytose synapses. So an alternative approach here would be to investigate whether astrocytes are similarly contributing to synapse elimination after injury. And if we do see alterations in GPR56 colocalization, be interested in using a knockout mouse in which GPR56 is specifically knocked out in microglial cells, and versus evaluating these mice using the fair conditioning paradigm and Golgi analysis, followed by the Morris-Water maze as a secondary measure if we see significant changes. And my expected outcome here is that the GPR56 knockout mice will have preserved dendritic spine density following a CCI, and that this would correlate to improvements in behavior. An alternative receptor that could also be implicated is TREM2, which is a receptor also known to bind phosphatidylserine and contribute to synaptic pruning. However, preliminary data suggested that TREM2 is decreased in our model, so we'll not initially focus on this receptor. So while we expect that microglial D-serine contributes to synaptic loss through this targeted complement-mediated approach, we also can exclude the fact that it may be contributing to global inflammatory responses. So this here is from a single cell sequencing data set in which mice were treated with LPS, an inflammatory compound, and 24 hours later, microglial cells were sequenced. And so I performed a secondary analysis here to look at some of our proteins of interest and saw that after LPS stimulation, there is an increase in serum racemase expression in microglia, as well as increased expression in SLC1A4, which is a microglial transporter of D-serine, which contributes to the release of D-serine by microglia. So while it appears that inflammation does induce serum racemase expression, it's unclear whether microglial D-serine synthesis can have the opposite effect, where it contributes to inflammatory responses. So to look at this, I'll be using a magnetic bead panel to look at the expression of different inflammatory molecules in the hippocampus and seeing if there are any changes when serum racemase is ablated in microglia. So as I brought up before, here we anticipate that microglial D-serine synthesis has more of a targeted effect, where it is damaging individual synapses, leading to their pruning. And we don't expect to necessarily see a change in global inflammatory response. However, it is possible that microglial D-serine is having an autocrine effect, where it can activate NMDARS receptors on microglia cells, contributing to increased secretion of cytokines and chemokines. So in this case, we would like to look at this using cultured microglia that are isolated from either control or knockout mice, and seeing if there is a difference in inflammatory molecules. To further look at this on a global level, I'm planning to use single cell RNA sequencing, where we will collect all cell types in the hippocampus and sequence individual cells to see if maybe there are specific subtypes of microglia that are upregulating serum racemase after injury. And in using the microglial serine racemase knockout mice as well, I'm hoping that this will help evaluate the contribution of D-serine on heterogeneous injury responses and allow us to identify downstream signaling effects of D-serine on various cell types after injury. So the third aspect of my project is focused on evaluating these components that we've identified in our mouse model and seeing if these are present in human TBI cases as well, where we've been collaborating with Dr. Gracioli, who is a neurotrauma surgeon here at the University of Miami Jackson Health System. And he has been providing us with tissues that are resected from patients undergoing neurosurgery that have suffered a traumatic brain injury. So here we are receiving acutely injured perilesional tissues. And I'm planning to evaluate the presence of D-serine induced pathology in our own CCI model and seeing if these are present in the human patients as well. So I'm specifically focusing on some of our key proteins of interest, such as serine racemase and the D-serine transporters like 1A4, as well as the complement proteins and NMDA receptor components. And I'm planning to also look at this on an RNA level using RNA sequencing, as well as on a protein level. So just to show you some of the patients that we've processed so far. We have processed 12 samples, where, as you can see, we have a variety of injury types caused by falls or motor vehicle accidents, as well as a couple of gunshot wounds. And we receive information about the patient's demographic information, such as their age and sex, as well as race. And we also receive information about the severity of injury at the time of arrival. And we'll be receiving information from follow-up visits, so we can eventually see if serine racemase expressions correlate to functional outcomes. So some of the preliminary analysis that I did shows kind of this bimodal expression of serine racemase after injury, where it seems that half of our patients have higher levels of serine racemase expression, whereas some show no change at all. So in one way of thinking about this, we're trying to categorize this based on the severity of injury, where it seems that patients with more severe injuries might have an increased expression of serine racemase. Another way of categorizing this is to look at patients based on the area of injury, as there could be heterogeneous expression levels in different brain areas. So most of the patients we've received so far have had injuries to either the frontal or temporal lobe. And we're also interested in looking at this based on the time after arrival to surgery to see if the time after injury also correlates with expression levels, where it seems that perhaps longer, that patients start to express higher levels with more time after injury. And I've also looked at some of the other components that we're interested in, such as C3, which is significantly upregulated in TBI patients, where it seems that more severe patients express higher levels of C3. And when looking at this from a temporal aspect as well, it seems that at longer times post-injury, patients express higher levels of the C3 protein. And I was only able to analyze GPR56 expression in a few patients so far, but this also appears to be heavily upregulated after injury. So I'm planning to continue evaluating patients and processing tissues as they come in, and looking at this as well on a global level using sequencing techniques. Okay, so in conclusion, what we know so far is that the ablation of microglial serine racemase is sufficient to preserve synapses in the hippocampus and rescue behavior, specifically on a fear conditioning paradigm. And pharmacological inhibition of the GluN2B subunit of NMDA receptors was also sufficient to preserve dendritic spine morphology at 3 days post-injury. I've also observed increases in the complement protein C1q in our control mice, which is abolished in the absence of microglial D-serine. So some of the future directions, as I mentioned before, is to look into this gluN2B signaling pathway and determine whether microglial D-serine is enacting synaptic damage through the hyperactivation of extrasynaptic glutamatergic receptors, and determine whether complement is involved in synaptic pruning after injury. Also going to be evaluating this in human patients and looking to generate RNA sequencing data sets for these patients. So with that, I'd like to say thank you to everyone, especially Dan, my mentor, who's always so supportive and helpful with interpreting all the data. Of course, Stephen, who just graduated yesterday, for passing on such an amazing project and helping me do everything and learning how to do everything in the lab. Madeline, who's a postdoc in our lab, is always very supportive and especially helpful with processing the human patients. And of course, a huge thank you to Jose, Maria, and Magda for everything they do, taking care of animals and performing surgeries, as well as technical support. I really couldn't have done anything without their help. And of course, shout out to Maureen, who's also in the lab with me. And thank you to all our collaborators and all of you for listening.
I thought that was a very nice presentation. Congratulations on putting it all together. And it looks like it's going to be a fantastic PhD thesis. So all the best for the future. I was interested in the regional distribution of some of these changes, and most of your mouse studies, I would assume, are going to be focusing on the hippocampus, but then you jump to the human studies where there'll be, I assume, mostly cortical tissues. So do you think there may be some differences in regional distribution of some of the targets you're looking at?
Yeah, I think that's definitely possible, and something that I've thought about in terms of translating this to humans. So yeah, so far everything we've looked at has been in the hippocampus, but I am working on collecting cortical samples from mice to see if some of these mechanisms are also present in the cortex as well. And then we're also planning to maybe receive some subcortical areas as well, because in speaking with Dr. Gracioli, he said that when they insert cannulas into patients to monitor intracranial pressure, sometimes they receive subcortical tissue as well when they remove this cannula. So that's something that we'll also start receiving soon to hopefully try to evaluate some of this on a subcortical level as well.
Very good. Thank you very much.
Thank you.
And I have a quick question. It's more technically related. You're proposing to use Golgi analysis to look at synaptic changes. I've been hit by a reviewer's comment saying that this technique is problematic and that it's just arbitrarily selecting a subset of neurons that might be biased in some way. What other approach are you thinking or considering to use to maybe deal with that bias, to look at synaptic loss and with a different technique that maybe doesn't have these issues with Golgi analysis?
Yeah, so previously the lab has used electron microscopy to look at synaptic density in the hippocampus, which is something that I could use perhaps as an alternative to the Golgi stain. But yeah, I agree that it is randomly staining cells, but yeah, hopefully, I mean, I could look at some electron microscopy to try to correlate it with Golgi. And then I think that, I mean, if we're also seeing improvements with long-term potentiation and behavior, then that's a stronger case that this Golgi analysis that we're seeing is representative of the entirety of the hippocampus.
Are you looking at male and female animals? I missed that, I'm sorry.
Yeah, so everything that has been done in the lab so far has focused on male mice, but I am also evaluating the females currently. So I haven't had a complete data set yet, but I've been looking at fear conditioning in female mice. And so far, it seems that there is not this deficit on the fear conditioning task in the female mice, in either the control or the knockout mice. So I think that, I mean, pretty soon I'll be looking at actual expression of serine racemase in microglia in female mice. I know a lot of studies have shown that female microglia are less activated than male microglia. So perhaps the female mice don't upregulate serine racemase in the same way. But yeah, that's definitely something I'll be evaluating moving forward.
Yeah, that's an interesting observation. So take the ovaries out of the mice about a week before you produce the trauma, and you'll probably demonstrate what you're showing in the male rats. So that's a lot of studies that have been done on gender differences in TBI. It's a pretty hot area, and it's a little controversial, but your results are pretty consistent with what we and others have shown when the animals are very young. So anyway.
I guess if there's no more questions, thank you. Great job.
Thank you, everyone. Thanks so much for attending, for your questions.